I will be using a diploid eukaryotic organism to explain and illustrate linkage and three-point mapping. For this example, I will be using a canine breed of Dalmatians. The three loci that I will be discussing are linked and can express different alleles. Looking at the alleles, you'll see that the dominant traits are on the left and the recessive are on the right. Starting with the parents, we can see that the mother is purebred for no dots, black nose, and green eyes, whereas the father is purebred for dots, pink nose, and blue eyes. The F1 hybrid is heterozygous. It displays all of the dominant traits but also carries the recessive alleles. This is the gene map of the F1 heterozygous individual's chromosomes. The distance between each individual loci influences the probability of crossing over. If a crossover event does occur, the gamete produced would differ from the parental generation. The distance between the locus for the nose color and dot presence or absence is 5 centimorgans, and the distance between the locus for dot presence or absence and eye color is 12 centimorgans. The coefficient of coincidence is 0.83, making the interference 0.17. I will use this gene map to predict the probabilities of these possible gametes. The no crossover event gametes will resemble their parents and have a probability of 41.7%. The two gametes that will be formed from a double crossover event will have probabilities of 0.2%. We can see that the probability of gametes from this event of a single crossover at the first region is quite low at 0.2%. This is because the two loci in region 1 are very close to each other, which makes crossing over less likely to occur. Here are two examples of calculating the probabilities by using values such as R1, R2, C, and DCO events. We can see that the probability of SCO2 gametes is much higher than it was for the SCO1 probability. This is probably because there is a greater distance in region 2 between the two loci, making crossing over more likely to occur, with a probability of 5.7%. I've created an analogy to help illustrate linkage and the concept of crossing over. Here's the F1 heterozygous individual's chromosomes on a G map. I'd like for you to imagine that the parental chromosomes are separate highways. These highways also have exits, and these exits create connections between the two highways. The exits will be seen as crossover events. Traits or alleles will be seen as different destinations, such as gas, coffee, and burgers. A non-crossover event will be visualized as two separate cars driving down the parental highway chromosomes. Neither of the cars will take any exits, meaning they do not cross over to receive any food or gas from the opposite highway. In a single crossover event in the first region of the chromosome, we can see that the two cars both take the first exit of their highway. So each car received burgers from their original highway, but both received gas and coffee from the opposite highway. In a single crossover event in the second region, we can see that both the cars wait to take the second exit in their highway, meaning both cars receive burgers and coffee from their original highway, but both cars also receive gas from gas stations on their opposite highways. In a double crossover, each car would get burgers and gas from their respective highway. Both cars will receive coffee from their opposite highway by taking an exit or crossover, but both cars will return to their respective highway or chromosome to receive gas from their original highway, meaning both cars will take two exits or cross over twice in order to get to their destination or obtain an allele.